in this afternoon session, we're going to discuss very much detail about fundings of the OPTIMA project. So this morning it was a very interesting session. There were like a plenary session for the 3 projects, and we had 220 participants actually. So it was quite impressive to be so many, and also many from the European Commission. So that had been quite nice, and it was a lot of also interaction, many questions, and you know, interesting. Let's say concern. So today I'm very pleased to have in this panel 3 very well-known and very good researchers in the specific area, and that they would present different subjects and different topics of the OPTIMA project. And these are my colleague from Agricultural University of Athens, Professor Tsitsigiannis, who is going to start, and then it's going to be the next one. It's going to be Gerrit Polder from Wageningen Research, who is going to talk about detection, models, and prediction. And then the last but not least, very well known the area of sprayer application, Professor Emilio Gil from University Polytechnical of Catalonia in Barcelona. So for the ones who didn't attend the morning session, just want to say that OPTIMA actually runs for 3 years already and a half, and it's going to finalize in June. And then just very briefly to say that OPTIMA actually, just one slide and then it's going to stop, is working on prediction, have a decision support system on detection, advanced disease detection, selection of the PVPs and BioPVPs application and assess. So this afternoon session, we thought to be dedicated to the main aspects, and because we have the more results, it's going to be mostly prediction and detection, which is going to be covered by Gerrit. Selection is going to be covered by Dimitris, and then application is going to be covered by Emilio. So having said that, I'm very pleased now to call on mic, not on floor, but here on microphone, my good colleague and friend Dimitris Tsitsigiannis, who is going to present the first talk. Dimitris, the mic is yours. And please, just a practical question, everybody who may have some questions, please put them in the chat, and we will try to have some questions in the end of each presentation, 5 minutes, and then final questions in the end. Thank you very much.
Thank you very much, Spyros, for the introduction. So I will focus on the IPM system of the OPTIMA project. How can I move this from here? Anyway, so of course I know that you all know what is an IPM, but I would like to point out that IPM is not only the use of pesticides and biological control products. It's basically a framework of many different actions that a lot of them are actually OPTIMA is taking into consideration in the different work packages. So the focus of the work package 3 that I'm leading is basically to develop novel, sustainable, integrated pest management strategies that basically can be applied as preventive or therapeutic control methods for 3 selected diseases that I will talk about: downy mildew in grapes, apple scab, and alternaria leaf blight. So, and then several, we see biological and chemical intervention strategies were actually tested during the project in order to expand the range of available tools that we have, and the best performing option then were optimized in order to apply it in the field in a pilot test. So basically the objective were to evaluate a collection of many different categories of commercial and under development biological plant protection products and plant-resistant inducers. So we focused more on commercial plant protection products because those can be easily basically applied and immediately in the field. Of course, also we tested and screened new generation commercial synthetic PPPs. And another aspect also that we took into consideration was to understand basically the genetic, evolutionary, and agronomic drivers of frost resistance and the evolution of plant protection pathogen resistance. And finally, basically to design the optimum IPM strategy for the different cases. So why we focus on biopesticides? Because as you can see from this graph, the trend actually in the market is going quite high, quite up in the next years, and this is because a lot of very successful synthetic pesticides will be withdrawn or are being withdrawn continuously, and then we don't have any. We will not have a lot of the pesticide that we have. So there is basically a trend of many labs now and a lot of the industry targeting in the development of new biopesticides. So why we selected these three diseases? First of all, grape downy mildew is a major disease of grapes. It's a very serious threat in all over the world. The disease, as you can see, can affect basically all the green parts of the plant. And the management is basically focused on the use of fungicides that basically prescribed by weather-based warning systems that collect information on temperature, rainfall, leaf wetness, and humidity, and then can be applied. Alternatives, though, to chemical treatments, such as the use of biocontrol agents or resistant cultivars, currently play a really marginal role in controlling the disease. And also, Plasmopara viticola is a fungus that can penetrate the tissues. And once can penetrate and enters the plant, is extremely difficult, basically, to stop the disease. And then we can use systemic fungicide that can basically partially stop the disease. The other pathogen is Ventura inequalis that causing apple scab, another really important, I would say maybe the most important apple disease that can affect a lot of the cultivars. And also the control here is mainly based on the use of chemical fungicides with application of about 15 treatments of synthetic fungicide per year average. Also here, the application timing of fungicide has been strongly implemented using usually weather forecasting and disease forecasting modules. And curative application usually are carried out in critical situation under prolonged wetness, high inoculum amount, and with many different fungicides. So there is also here a really high need to optimize alternative strategies for the control of apple scab based maybe on natural substances or other inorganic compounds and biocontrol agents. The third disease is basically the alternarian leaf blight in carrots, another very serious diseases all over the regions that cultivate carrots. And it's caused by Alternaria jauki, is a foliar pathogen that can basically cause really epidemics in regions with moderate temperatures and the leaves are exposed to prolonged periods of wetness. And severe epidemics have been reported that can reduce basically the yields by 40 to 60%. And under high disease pressure, no single control measure is actually sufficient to manage the disease adequately. Also, in this case, the disease management is basically based on the application of synthetic plant protection products and partial resistant varieties. So, during the OPTIMA project, we focused in work package 3 on the finding products that are combining basically biological control products and chemical control products that can basically manage the disease. So before starting presenting some of the experiments, in order to apply a biocontrol agent, you need to understand the mode of action. That's really important in order to design the appropriate IPM scheme that you want for a culture, for a host. So the biopesticides, the mode of action, the biocontrol agents, for instance, the mode of action is based on the competition for space and nutrients. As you can see here, for instance, that we have a biocontrol agent, a very well-known is Trichoderma hertzianum, that basically is competing a pathogen that is Fusarium and can basically, as you can see here, they can leave no space for the for this particular fungus to grow, the pathogen. Also, could you please turn off your microphones? Also, another mode of action is basically the production of enzymes by the biocontrol agents, such as here also Trichoderma, that you can see here. This is the hive of Trichoderma that produces enzymes that create holes basically in the pathogen and can, in this way, can kill it. Also, there are a lot of biocontrol agents that produce antibiotics, as you can see here. So this is one of the biocontrol agent, and this is a pythium, a pathogen that cannot grow because the biocontrol agent produce antibiotics. And another also mode of action, a very important, is the induced resistance. So when we apply a pathogen, a biocontrol agent, for instance, on the leaf or in the root, can basically induce the resistant mechanism, but different molecules in the plant, and induce the systemic resistance of the plants. That's something really, really important to have it in mind. So with all this in mind, we started basically screening for each basically plant, for each host and for each disease, different bio-PPPs and synthetic PPPs in a 3-4 year now period, because we have an extension in the project. And basically during the first year, we did a lot of experiments in France, in Italy, in Greece, under greenhouse conditions with selected basically pathogens. And then we moved during the second year in this under semi-field conditions. So the best products that we selected under greenhouse conditions, we tested under experimental field condition, under semi-field, as we say, conditions. And then finally, during the third and now in the fourth year, the best combination for each particular crop and disease are being tested under commercial field and using also the novel spray system and advanced detection techniques that the next speakers will talk about. So first of all, how we evaluate the different, for instance, biocontrol agents. So under greenhouse conditions, for instance, we prepare the plants here, just so alternaria plants in a greenhouse in France, and then we spray the bio-PPPs under either once or 3 times, depending on the experiment. And then we covered, especially this one, to have high humidity for a few days, and then we evaluate basically the disease. How we evaluate the disease, I will show you in a bit. So here in an experiment that we have we did it in greenhouse in Greece, selecting different biocontrol agents and plant-resistant inducers using 2 varieties, and basically using a scoring system from 0 that has symptomless plant, 9 that basically the plants are totally blighted, as you can see here. This is the susceptible, and this is 0, no symptoms. So this is the scale basically that we use, and this is also for all the cases. Here is a list of different biocontrol agents and plant-resistant compounds that we used. And here you see basically the disease progress curves, the disease severity of the plant over time, okay? And this is the control, no treatment. See, and here you see the different compounds, how they behave. And just to show you here that basically a disease severity scale that is basically a disease index of 3.4 is considered below this particular score. Plant breeders consider highly resistant for this particular pathogen and the crop, for carrot, for instance. So here you can see basically for the different compounds that we checked, the performance, the total amount of disease. So this is the control, and here you can see the biocontrol agent. Of course, you can see here that none of the biocontrol agent is able to reduce 100% the disease. Okay, so here you just see from 50 to 70 to 80% decrease basically in the amount of disease. And you can see here, this is one particular cultivar that is more susceptible, and the other one that's a bit less susceptible. Again, the evaluation during trial 1 for the different bio-PPPs. And here, then we move to a second trial in greenhouse, and then we start applying more pathogen and performing more applications. So here you see basically 3 different applications of the same bio-PPP. So in after 15 days, different 10 days intervals, and then 3 times spray with the pathogen. So we want to see how the different bio-PPPs can basically behave when you have really high disease pressure from the pathogen. And again, you see the two different cultivars, and you see also the differentiation of the bio-PPPs. And something that is really important that we saw it in all the cases and in all countries, that the bio-PPPs are not able to behave in the same way in the different cultivars. So maybe one bio-PPP, a specific bio-PPP, can work very well in a cultivar, but in another cultivar might not be working at all. As for instance, this one here, the green one here, can reduce the disease, but in this particular cultivar is not able to reduce the disease. So basically, the carrot experiment were done in Greece and France, and we found quite a lot that were basically quite efficient, and but the efficiency is depending on the genotype. Again, similar experiments were performed in Greece and Italy for grape downy mildew, checking 2 different cultivars in Greece, another 2 cultivars in Italy. Italians did a lot of experiments also under field conditions, using this particular disease index that to measure basically the disease. And here you see a list of the many different bio-PPPs and resistant inducers that can that have been applied. You see again the similar results on in Greece and different cultivars under low disease pressure, and here also under low disease pressure. But in Italy, when they tasted a lot of bio-PPPs under more than 15 under field conditions and under very high disease pressure, almost 100% of leaves were covered with downy mildew, then none of the bio-PPPs were successful in controlling the disease even after 11 treatments of the same bio-PPPs. So under high disease pressure, then it's impossible to control it with only bio-PPPs. And same experimentation also was performed under greenhouse and field condition for the apple scab using different bio-PPPs. So the active compounds of bio-PPPs, as I showed you, are very different, and as a result, dissimilar mechanisms of action are expected, and these mechanisms seem to be diverse in different cultivars. So combination of several bio-PPPs are, in order to increase the level of protection, is really critical. Also, we tested for the different pathosystems, many different new generation fungicides. So the fungicides were quite efficient in controlling the disease, especially in Alternaria and carrot. You can see here the effectiveness of several fungicides, and in different cultivars. Fortunately, with the fungicides, we don't have the phenomenon that we saw with the bio-PPPs, that different cultivars behave, the bio-PPPs were behaving differently. Here, the fungicides were quite effective. Most of them independently of the cultivar that we were using and the disease pressure that we were basically using. And they were quite effective for a very long time. And again, for the downy mildew, we had our colleagues in Italy did similar experiments, and they found quite a lot of fungicides that could actually, new fungicides that could control nicely the the pathogen. So then we wanted to see basically how the host resistance and in combination with the BioPPs can be altered. What we mean? So we wanted to evaluate basically using different systems, especially transcriptomics approach, how the different BioPPs and if the different BioPPs can activate the defense system of the plant. So we used a novel approach that our colleagues in France had developed initially for apple and apple scab and downy mildew, that basically is the simultaneously expression of 28 targeted genes that are being involved in plant defense system. So at the same time, so what you are doing, you apply basically the BioPPP and you check many different genes that are involved in several biochemical pathways, as you can see here. Has been stuck.
Yes, we hear you, Dimitris. Yes, it's clear. Please.
Yes, but I cannot move forward. Okay, now. So as you can see here, this is the experimental approach. We spray basically the BioPPPs on the plant. We extract RNA, and then using a plate, we can at the same time basically check the expression level of many different genes. So here you see basically 11, for instance, this is apple scab. For apple scab, 11 different plant-resistant inducers that have been applied on apple. And you see, for instance, this one, C, that's quite red. That means that many genes are quite highly induced by this particular inducer. So that means that this particular plant-resistant inducer can nicely activate the defense system of the plant. So we also, in Greece, we selected the best, based on our previous experiments, the best 3 BioPPPs and pathogen-resistant inducers. We applied in the field and 5 times, and then we collected samples before the pathogen infection in this point and after the pathogen infection in order actually to identify if genes and which genes are basically can be activated. So you can see here all these green, red genes are the ones that basically we check it can be activated by a specific BioPPP pathogen-resistant inducer. And here, basically, you see the different cultivars that we did the experiments, Pest and Polder. This is a very susceptible. This is a moderately partial resistant. So you see the expression of all the genes, and here the different BioPPPs. And the analysis, what they saw basically is that 2 of the BioPPPs, the BioPPP 1 and 3 basically, that work quite nicely in controlling alternaria, they also induce many genes that are involved either in early responses of the defense mechanism or the phytohormones, PR genes, phytoalexins. On the other side, the BioPPP2 didn't work at all in Presto to control alternaria, and this particular compound, BioPPP2, is not able to activate basically any of the...
foto, pero me ha dicho Alberto que se estaba muy bien.
Could you please turn off your microphone?
I did it.
Okay, thank you.
Yes.
So the other cultivar, the Bolero, here all the BioPPPs basically were working quite well. And here we see exactly the same, that we have quite good induction of most of the genes in this particular host. Besides the transcriptomics approach, we also wanted, we are currently analyzing metabolites that are being induced after after the application of bio-PPPs, because we also want to see if specific metabolites that are involved in a defense mechanism such as terpenoids, phenylpropanoids, and other compounds can potentially involve in GARD resistance. Okay, so another aspect for the resistance has to do also with the fungicide resistance of the particular pathogens. So, for instance, with Alternaria, we checked if many different isolates from all over the world are able basically to still be resistant with the new selection of the best fungicide. So the best fungicide is still working under different resistance. So and we were able to identify that basically all the tested isolates were quite resistant, quite susceptible to the selected basically fungicide. So finalizing, the last step is now to design the IPM strategy. So I will use the Caro Alternaria and Carrot as an example, and basically how we designed, how we selected now the PPPs in order to identify the optimal IPM strategy that combines also dosage, formulation, timing of the application, and all this stuff. So in carrots, for instance, for Alternaria, based on experiments in France and in Greece, we selected several bio-PPPs and several synthetic fungicides, and we designed 20 different IPM schemes with the different combinations, alternating also the bio-PPPs and synthetic PPPs. And here you see, for instance, the results for the one variety, the Presto variety, that you see the different IPM schemes here. Okay, and combination of 3 biopesticides with 2 fungicides, 3 biopesticides, 3 fungicides, only biopesticides, fungicides, and so on. And also we had what farmers basically are currently doing as a control in Greece and France. So this is basically in Presto. This is the 3 bio-PPPs that we tested also with the transcription of the 28 genes. So, and also this is the one that I was telling you before. This one, the bio-PPP2, as you can see here, doesn't control the disease. You see, it's the same like the control plant, doesn't manage the disease. So this was not able to actually activate the defense mechanism of the plant in this particular cultivar. On the other hand, if we move now to the next cultivar, which is Bolero, here we see that this particular bio-PPP is working. So in this particular cultivar, is able to induce also the defense mechanism. So again, you see also Bolero that all the IPM schemes that we tested were able to control the disease. However, I have to say here that we didn't have really, really high disease pressure under field conditions here. Of course, for the IPM, we tested also the plant residues in order to be under to be below the MRLs. So the optimal IPM route that resulted from all the previous tasks has basically started from task 3.1 that we concluded basically that a route that is exclusively based on bio-PPPs is not realistic under high disease pressure. It's not, the protection is not good. Then new generation PPPs allow a very good protection of several of the diseases, and identification of the optimum bio-PPPs with a QPFD analysis that we did. Then a mixing route, including the best bio-PPPs and alternated with a new generation of synthetic PPPs. And then for Alternaria, for instance, we finally decided to combine one new generation synthetic fungicide, then a bio-PPP, then another fungicide, and finally a plant-resistant inducer. And if the disease pressure is high, we can also finalize the IPM scheme with the addition of another extra bio-PPP compound. So this is the route that we selected for Alternaria, for instance, that was applied also in France, that Emilio later on will show you the some of the treatments there, and the cultivation week that the product was sprayed, and so on. So based now on all the information that I saw you with the selection of the best IPM scheme, and you will see in the next two talks how we can also able to predict the disease, how we can diagnose the disease with new means in order to identify, in order to define, sorry, a disease risk map, as you can see here, for instance, and then apply the different bio-PPPs and the synthetic PPPs in order to have a holistic IPM approach to manage the disease. Thank you very much for your attention.
Thank you very much, Dimitris. Very impressive results. We all know within the project how much work you and your colleagues from UNITO and INRA have put on this work. And I think many people are very interested in bio-PPPs, of course, and the combination. So there is a lot of attention towards this area. Are there any questions, please? If there are any questions, you can just put it in the chat, and or otherwise we could have a discussion in the end.
Mm-hmm.
Okay, so just a question, Dimitris, from my side is like, because people are waiting to see like these IPM strategies for the 3 crops. I mean, the combination of synthetic and biological PPPs. So in practice, if you were if I was a farmer and you were the researcher, would you advise me? I mean, how many, and I was applying, as you say, for Apple's Cup 15 on average on your presentation 15 times a year. How many times would you think it could be replaced by a PPP, bio-PPPs, biological,
yes,
and to be also effective?
Mm-hmm. So I think based on what we see, you have basically to start with the synthetic PPPs for preventive purposes, to be sure that you don't have the fungus enter the plant. So it's very critical to apply the best chemical, or maybe if you have a good bio-PPP at the very early stages, but I suggest to start with 1 or 2 doses of synthetic at the beginning, preventively, and then maybe try to go a little bit more slowly. Then you can apply alternatively 1 bio-PPP and 1 synthetic one. For downy mildew, though, the situation is much more difficult because it's a really difficult disease to control. And here we need a little bit more experimentation to define if we can really apply bio-PPPs at the beginning or when we have to apply the. So, but we will have this data, I think, by the end of the project.
Exactly, and we have promised that we're going to have like very good guidelines and like a protocols how this can be done. So that will be very interesting with the project, and we'll probably do also some video presenting some of these results to be more applicable.
Yes.
Again, thank you so much, Dimitris and your colleagues. It's
thank
excellent work you've done for this part of the.
Yeah, that was a great teamwork, actually.
Definitely, but and just to say to everybody that it has been so hard, as you can imagine, during the COVID, the last 2 years.
Exactly. To do the experimentation under COVID conditions, extremely difficult.
Yeah, for example, I know, for example, in Agricultural University of Athens for 6 months, it was only Dimitris and his team was coming to the university to finalize or do the results for the OPTIMA project. So thank you very much for this contribution anyway. So again, thank you, Dimitris. We'll come back to you with the questions and answers in the end. So we move to Gerrit Polder from Wageningen University for the next presentation. Gerrit, the mic is yours.
Yeah, thanks, Spyros. And Dimitris, also thanks for the nice presentation. Very nice overview and insight. So let me try to start my share my screen, which is this one. Yeah, and try to. Okay. So you see my screen now?
Not yet.
Not yet. Share. Now you see my screen.
Yes, clear.
Okay,
Yeah.
great. So I like to present another work package from the OPTIMA project. The title is The Role of Prediction Models and Detection Systems for Early Identification of the Diseases. The work I present is a combination of what we did in Wageningen with my colleagues Pieter Blok, Jozef Pell and Tim van Dalen. The work of Argenzo with Zisis Tsiropoulos and his colleagues. But I have to say that also the other partners in the project did quite a lot of work for us, also due to COVID, where we were not able to visit the trial fields. And a lot of images were acquired by the partners and also annotations. So let's start. So very briefly, Wageningen University and Research introduction. We are a research and university organization working with industry, governmental authorities and other knowledge institutes. We have about 6,000 employees and 12,000 students, and we are very international. Maybe most of the audience already know about Wageningen. We have students coming from 100 countries all over the world. We have a group working on research in agro-food robotics, where I'm also part of, which is a group of about 65 people. So for myself, I work quite some time already in Wageningen, more than 30 years now, as a senior scientist in computer vision for plant phenotyping, specifically using spectral imaging, and currently working quite some projects in disease detection. Actually, my background is electronics and applied physics, and I'm the OPTIMA work package tool leader. So here are the main objectives from OPTIMA. This already presented by Spyros this morning. The focus of this presentation is on the models and the development of the advanced early disease detection methods. As already also mentioned before, we have 3 trials in France, Spain, and Italy with 3 different crops and 3 different diseases. And we worked in all 3 of them with our detection system and with the decision support system. So how does the system looks like, what we developed in the end? It's a combination of the early detection system and the decision support system. So we developed a camera-based early detection system, and this is an example for apple scab, but the same we did for carrots and grapes. This smart camera system detects the disease in the field, and they are sent with information about the location to the decision support system developed by Argenzo in the cloud. So there is a cloud system running, and we send our detections and location information to the cloud. And there the decision support system analyze the data together with a lot of other data, like the weather data, historical data, the geographical data, etc. And then it will give an advice on the spraying actions by providing prescription maps. And here you see a short video of the DSS system in operation. So this is a geospatial interpolation for the risk for the next the next 5 days, based on all the information acquired by the DSS. So this information is provided online, so you can use it from a browser. You can also use it from a smartphone. And then the system also gives prescription maps based on the outcome of the early detection system and the models behind in the decision support system. So here you see 3 examples of prescription maps. 2 of them are in apple and 1 of them is in grapes. The most right one is grape. And this prescription maps is then loaded into the sprayer, and the sprayer sprays where the risk for the disease is high. So then switching to the early detection system, we worked actually on two different systems. We worked on an RGB imaging system with a smart camera. We worked on spectral imaging. The RGB imaging uses deep learning as artificial intelligence technique to detect the disease from the images. And for the spectral imaging, we used traditional machine learning earlier, but in the end, we also use deep learning to make the predictions. So let's first focus on the RGB imaging. During the course of the project, several cameras were used. So we started with a camera rig with some spectral cameras, but also with an RGB camera, and that's the IDS camera, which you see here on the left. It's an industrial camera with quite a high resolution, 10 megapixel. Then, since we were not able to visit the field a couple of times due to COVID, also the partners made a lot of images from the field, from the diseases using several smartphones. So those were used in the algorithms. And then in the end, we built this smart camera system using a smart camera with a AdLink Neon. The type is the AdLink Neon 2000. It uses a NVIDIA graphical interface and GPU. And the camera used was, we have two cameras actually. We have three systems with two different cameras, and 1.2 megapixel and a 1.9 megapixel camera. So quite a lot of different cameras used in the project. Then the system we developed in the end for using in practice, the smart camera system. Here you see an overview how this works, how this looks like. So we see, let me put on the laser pointer. So here you see the smart camera system, so the camera and the GPU. But we need more to do the detections. We need a connection, of course, to the cloud, but we also need information about the location, and therefore we need GPS. So we have this combined GPS cell phone Wi-Fi camera antenna, which is connected to a router. And all this information, to send out the information to the DSS and to grab location information for the GPS, is connected using USB to a docking station here to the camera. And then, of course, it's important for the user as well to have some feedback. So there is also a screen connected to the system. And since we like to acquire also the images to do retraining in the end, we added a solid state hard drive to do the recordings of the images. And there is some power management in order to run the system from a battery. So here is a picture of the system, how we developed it in the lab. So the smart camera, the router, and the USB controller, and the power supply, etc. And here you see how the system looks like in the field. So this is in Spain. So we have this small trolley behind the tractor. Here's the battery. And then here we have the smart camera with the touch screen here. The GPS antenna on top. And this is the box with the other electronics. And here you see another view of the system, how it looks like. Here is a picture of the screen of the system. So this is what the user of the system sees when the system runs. So here you see the large part of the screen is just an image of the camera with the detections. Here you see 2 Downey mildew detections. And then here you see other information like the time, the GPS time. So this is the exact time from the satellite. The quality of the GPS based on the number of GPS, the number of satellites, and the signal strength, and then the latitude and the longitude. So this is the location information. It detects whether the system is moving or not, and then also some information about the system like memory and images and etc. And then here in the bottom line, we see we have 2 hits, and the probability is 0.56 for the hits, for the detections. Yeah, as I already said, we used deep learning. Let me start this video. Yeah, so we used deep learning for the detections, and I first like to explain a little bit about deep learning approaches. There are different approaches for deep learning. You can do classification, then you put in an image, and the algorithm classifies the image in a certain class. So if you hear an image of a cat, then this image is classified as a cat here properly, and if it's classified as something other than it's a wrong classification. We can also do object detection, and there we work with bounding boxes. So within the image, there are bounding boxes around objects which are detected. So here we have an object detection of a cat and the 2 detections of a dog. And you can also do instance segmentation, that it's not only the detection of the object, but also segmentation against the background and against the other objects in the image. What we used in our research within OPTIMA is classification and object detection. And we used several systems. We tested in the smart camera 3 different systems. The classification using efficient network. And there we classified between healthy and disease. So just an image classifier, it's 1 hit. The probability is then the confidence of the detection. The second method was detection. So this one, object detection, using a YOLO V5 network. And there we detect multiple spots in 1 image. And you see that in the video on the right side here. You see here the video when the device was running in the field, and you see the detections of downy mildew here in the grapes. And then the last method we tested was a fancy new thing network, also based on EfficientNet. But there we did classification on image patches. And you remember that we have a very high resolution camera. In the beginning it was 10 megapixel, but in the smart camera it was still up till 2 megapixel. But mainly the images used for deep learning have a much lower resolution. So therefore we just divided the image in smaller patches and did classification of each individual patch. Well, we tested them all in the field, and in the end, the detection network using YOLO V5 was the best performance, so we continued with that. And then I have to explain a little bit on the evaluation of the algorithm. So we came up with results, and how did we evaluate that? Well, we used an intersection over union to do the evaluation of the spots we detected. So we have these bounding boxes, and you have ground truth. So those bounding boxes are drawn by the crop experts, mainly the partners in Italy, France, and Spain. And then we have detections from the algorithm. So here you see in red the ground truth by the crop expert and the detection by the system. And if you have an bounding box which is not on the ground truth, then it's false positive, of course, and it's an error. If you have small overlap, then it's also counted as an error. And if you have a larger overlap, then it's counted as a true positive. And if it's complete overlap, then it's actually excellent. And then you have also a true positive. And since the spraying resolution is much lower than the image resolution, therefore we used a very low or a reasonably low threshold on the intersection over union. So if there is still here, this is a false positive, since the intersection over union is 0.11, but if you have 0.25, so a quart, then we count it as a true positive. So that's about the evaluation of the bounding box detections. And then how did we evaluate all the detections together? And here you see an example of how we did it with a confusion matrix. So we have the actual values, whether it's deceased or not, and we have what the predictions were from the crop experts. Sorry, with the prediction. So the actual values from the crop experts and the prediction from the network. If you have a proper prediction, then you have, of course, a true positive. It's a cat and it's detected as a cat. If it's not a cat, and it's the if it's a cat and it's not detected as a cat, it's the network says it's a dog, then it's an error. So that's a false negative. It's a type 2 error. And if it's not a cat and it's detected as a cat, then it's a false positive. And then it's a type 1 error. And we can measure those errors in a recall and a precision. And then we have to compare all the recall and precision values for the different approaches. And that's not not convenient to compare two values. And therefore we have a combined value and the F1 score. So you have 2 times the recall times the precision divided by the summation of the two. And note that we don't have these true negatives in the data because we don't have annotations of healthy spots. We only have annotations of diseased spots. But that doesn't matter for the F-score because in the F-score the true negatives are not used. So, yes, the F-score. Then I switched to the applications, the 3 trial fields. So first the downy mildew in grapes. We did quite some experiments in the past, in 2019, 2020, using the cameras from the IDS and smartphones. We had annotations from the crop experts, from the people in the field. And then based on that, we used the algorithm in the smart camera. But in the smart camera, also new images were acquired. And you see quite a lot of them, 70,000. A small portion of this is then annotated. There's 300 images as you see here. And those are used in retraining the algorithm. Let's go to the results. So here you see how the image is operating in the field. I think Spyros already showed this image this morning as well. And here you see some results from the network. And on the left side here, you see the system, the result, using the initial training. So this is based on the training of the 2019 and the 2020 data. Now you see quite a lot of errors. There's just one true positive, but we have a false negative. So a detection not found, and we have a false positive. But then based on the data, the new data, we did a retraining, and then you see we have completely 100% result for this single image with the true positives found. Here you see an overview of all the results. So this is in the trial field in 2021 based on the training in the previous years with quite some low score. But after retraining, the score is increased quite a lot. So the F1 score decreased. Actually, we did on the data of the previous years, we had high scores like 86 and 67%. But now, due to different image acquisition, the result was much lower. And that is since we did some very careful imaging in the previous years. And also the partners with the smartphones pointed to the leaves where specifically the diseases were. And made nice images of that. And now we are we have this smart camera on a tractor and we drive through the fields without looking how we take the images. So there's much more variety in the images. We have highlights, we have shadows, we have spots which are very small, you name it. So the message here is that if we have more annotated data, with more variation, then this score will be increased quite a lot. And I think we can come back to the scores which we have seen previously in 2019 and 2020. For Apple's Cap, the story is similar. Again, we have images taken in 2019, 2020. And we have a large number of images in the trial fields in 2021. We annotated 500 of them. And again, here you see the picture of the measurements in the field in Spain. And here you see very bad results on the original training. And yeah, main problem in this image is, as you can see, the highlights. And there's also, I think, copper sprayed on the plants, which also gives problems. So a lot of problems in the image here, but after retraining, it is quite a good job. 3 true positives, which are quite good, and this false negative, yeah, it's not a big issue because it's already connecting or overlapping a true positive. So for the spraying action, this is not an error at all. And here are the numbers. Very low in this case. In the previous years, it was much higher. And here the score is not that high as in the grapes, but it increased a lot from this. How we can increase this, that's a thing we come up, I will explain later. But first, let's show a little bit about the carrots. In the carrots, we had not that much data in the previous years due to several issues. But in the last year, we had quite a lot of them. And also a lot of them were annotated by our partners. Here you see an image of the system in the field during a expert training session. And here you see again, the image is one example image before and after training. And again, the results increased quite a lot. Just one error here, but still, if we make a spraying action here, then it's still not an error, this one. The numbers are here, very low on the pre-trained, but it increased up till 42. And in the previous years, it was also not that large. It was only 20%, mainly due to the lack of data in this case. And here I have to say the same thing, that we can increase this in another way. And the other way, that is spectral imaging. So this is the next part of my presentation. What is spectral imaging? Spectral imaging actually is a combination of spectroscopy and imaging. So we combine the two, and we have a spectral data here, which consists of a large number of bands, as we have the spectra here, of the images. So on each pixel, we have a spectrum. That's what spectral imaging is. You can also call it imaging spectroscopy or spectral imaging. And why do we do spectral imaging? Because imaging gives access to the plant structure, and the structure of the Z-spots, for instance. And spectroscopy provides information about the chemical composition. And if you look in the visible range, 400 to 700 nanometer, we're looking at pigments. And in the infrared, moisture and other internal structure of the plants. During the experiments in OPTIMA, we use several camera types. And here I have 4 modalities, which I briefly like to explain. If we do just plain spectroscopy, we don't have any spatial information. So it's actually not imaging. So this spectroscopy. But then we have the next one, which is a few number of bands, but very low spectral information, just 3 bands in this case. And this is, yeah, the same as with the color camera, just 3 bands. You can call a color camera also a spectral imager, but it's just in the red, green, and blue band in that case. If you have more bands, like here, then you have reasonably high spatial information and a moderate spectral information. And if you use a large number of bands, so like 200 bands or 300 bands, then we have a high spatial and also high spectral information, but this is slow. If you acquire those images, then it's very slow. So those are the cameras used in the experiments. So in the first year, in 2019, we did some field experiments and also greenhouse experiments using this IMEC SnapScan camera. And then, based on the results, we decided to buy a multispectral camera. That's this one, with a few number of bands, but it's fast. It can be used real time in the field. And this cannot be used real time in the field. So here you see our experiments in 2019. He's my colleague Joseph behind the the laptop to acquire the images acquired by those cameras. So here you see the IMAX snap scan, high resolution spectral imaging. This is the RGB color camera, high resolution RGB. And this is a multispectral camera, 6-band. And here you see the spectra from the apple scab and the healthy and the diseased ones for downy mildew here and alternaria here. So it's not much difference between healthy and infected spots, as you can see, but still you can use this information. And what we did in first instance is using the data from the 2019 experiment. We did a linear discriminant analysis. So that's what I call traditional machine learning. Using the spectra, and then based on the linear discriminant analysis, this gives weights, which shows the importance of the wavelengths used in the analysis. And here you see the weights used in the LDA. And as you can see, here is a large weight for downy mildew, around 550 nanometer, and here are some other bands here in the infrared. For alternaria, it's here, some bands here in the infrared, here a band in the green. And here as a list, here the best wavelengths are listed. Same for apple scab, here in the infrared, a large one. We have some in the green and in around green and blue here, something, and the red edge and the infrared as well. So here is a list of the best wavelengths. Now, based on this information, we selected the camera, and here you see the camera again. It's a Celios camera, CMS V4, with 8 wavelengths. It's an 8 wavelength multispectral camera, and here you see the wavelengths of the camera. And here you see the overlap of the wavelengths within the camera with the peaks from the weights from the analysis. So the wavelengths mentioned here in yellow, so those wavelengths, those are overlapping with the LDA weights from the different crops for both the for all 3 the crops, for alternaria, for downy, and for scab. Then here you see a very preliminary experiment taken in spring 2020. It's a single trial we did in the Netherlands itself. It was COVID time. We took 50 images. We imaged them, and this is with apple scab in the apple leaves, but it's a very early stage. And as you look here at the RGB image, you can hardly see it, or you can't see it at all, the small spots. And if you look at a false color image from the multispectral, you see clearly the spots here, and there, the scab spots. So the contrast is increased quite a lot from RGB. If you do the analysis here, you see the numbers, but it was a small experiment. So this is based on 4 images with a few number of spots, and the detection was quite good. Using YOLO in this case, but we just used 3 bands. We just used a green band here, a red band, and an infrared band. But our camera has 8 bands. So the question now is, what is the effect if we use the YOLO V5 network when using images with more channels, 6 and 9 in this case, compared to using 3 channels? You have to know that the YOLO V5 is a specifically deep learning network for RGB images, so it's just made for 3 bands. So we had to make some adaptations to the network, and that's what we actually did. I'm talking about 6 and 9 bands. So originally it was 3, then we did additional 3 bands coming up to 6, and then we did additional 3 bands coming up to 9. And the camera has 9 bands. I was telling you that it had 8 bands, and that's actually true. Here are the wavelengths, the filter characteristics of the 8 bands. But the camera has 1 additional channel, and that's a gray value channel. That's the purple line here. It gives information over the whole range of the spectrum. So those bands are all used in the analysis. So what I already said, the YOLO V5 was adapted to 6 and 9 channels. I didn't say that before, but YOLO V5 is mainly working with transfer learning. That means that the network is pre-trained on the large data set, and that's the COCO data set from Microsoft. That's just general images. And that was done once more now, but we duplicated the COCO weights for the additional channels. That's how it was implemented. So we had a COCO weights standard for the first 3-channel image, and then we duplicated the weights for the 6 and the 9-channel image. So here you see how this looks like. So we started with the RGI image, so red, green, and infrared. We could not do RGB because the B channel was not in the data. If we go back here, you see it starts at 500 nanometers, so there is no blue information in the multispectral camera. And then we did false color based on the weights from the LDA. So those are the most important ones, 2, 4, and 8 bands, and then we did 5, 1, and 6, and then in the end 7, 3, and 9, and 9 is the gray value channel. Yeah, we did the training on 80, validation on 14, and again a small test set of 4. That's how we did it. So we did RGI, just false color 1, 3 channels, false color 1 and false color 2, 6 channels, and then all channels. And here you see the results. So from the RGI image, we increased a bit with the F1 score from with the FC1, 0.48 was 0.46. But when we use FC1 and FC2 together, then it decreases already a bit. And for the 9 channel, it doesn't add, it's even more decreased. So we have to note that these are preliminary results. And more annotated data, of course, will increase the F1 scores, but probably selecting the right bands is more important than having more additional bands. So that's the main conclusion here. This is my final slide with the discussion. So disease detection in combination with the decision support system are key technologies for integrated pest management. I hope I have shown that in this presentation. The scores as shown in this presentation are at spot level. I've mentioned it several times. When we are going to spray, then it's at plant level or even larger in the carrot case, for instance. So then the scores are actually larger. Data is very important. So we need variation in the data, and all data needs to be annotated, which is quite laborious. And we need crop experts there as well. So what we can conclude also from this is that for downy, the RGB, for downy mildew in grapes, the RGB is sufficient. We don't expect that we can increase with multispectral, but for apple scab and carrot, the infrared channel contributes significantly to the results. And if we use a proper selection of bands, then the detection will also increase. So more bands are beneficial, but require a large annotated multispectral data set. So that's also an option which in theory is possible, that we don't use the COCO pre-trained weights, but train the network completely with our own data. But in that case, we read a large number, a huge number of data. So I don't think that is really feasible. It's time for questions.
Thank you very much, Gerrit. That has been very detailed information, and thank you very much for all the work we have done. Again, we have worked very hard during COVID. So we do have one question, actually, from Nicola Vicino about RGB imaging and detection. I see that images, example, the ones about downy mildew, looks quite zoomed. Are more camera needed to get the entire canopy? Can you please respond to that?
Yeah, that's a good question. Let me which image are you referring to? Is that let me see.
It's for the downy mildew.
Yeah, and I think this is this one. No, this is apple scab. This one. This is not quite focused, I would say. So you see,
yeah, Nicola, if you like, you can unmute and discuss.
Yes, can you hear me?
Yeah, I hear you.
Very well.
Yeah, I'm sorry, but I am at home for COVID reasons, so I'm not
Be well soon then.
100% of myself. No, I was asking about the images we are seeing now. They look a little zoomed. I think that to get the entire canopy, probably you will need more cameras.
No, the camera is on the trolley or on the tractor, so it drives through the field. And I think here you see quite a large number of the field. And if you see the video, I can show you the video. There is quite some overlap as well.
No, I mean in the from bottom to upper part. Usually a canopy can be also 2 meters high.
Oh yeah.
So here we are seeing, I think, 1 meters of a canopy in the vertical dimension.
Yeah, I think it's more, but yeah, and if your crop is really that high, you can add more cameras. But it's a choice, of course. You can add more smart cameras. That's not a big deal. But you can also, not acquire everything in the crop and use it statistically. If there is some disease there, you expect that there is more. And if you find something, then will certainly also be something in parts of the crop which is not imaged.
Okay. Then also another question, what about the advancement speed of the tractor, of the system? At what speed can it go to get a real-time elaboration of the images?
Yeah, it was quite fast. We can do, I don't have it in the presentation, but I think we can do 10 images per second, something like that.
Okay. So
yeah, that's what I remember. 10 images per second, so you can drive reasonably fast.
Yeah, okay.
Yeah.
Thank you very much.
You're welcome.
So I think we're going to take one more question and then we'll move to Emilio. So the question comes from Manuel Paisios. Do you think using an additional light source, perhaps adapted to the more relevant bands, would help detection?
Yeah. Ideally, you use artificial light, and then you have control, and then you can do better imaging. But we have decided not to use artificial light for those applications to make it easier to implement. So yeah, it can help, but I'm sure if you add in more variation by just acquiring more images and do training on them, that it will be the same result in the end.
Exactly. That was the decision to make it easier to be adapted afterwards in farming conditions by farmers.
Yeah.
Okay. So thank you very much, Gerrit. Another thing I want to say that thank you for also showing the cats and the dogs that make everybody, realize what AI. So what's the relation between dog, cats, and diseases? We can have AI for identifying the diseases or the cats or the dogs. So that was very good, actually. You gave it to present it. So thank you very much again for your great contribution to this project. And then we move to Emilio Gil, who is going to entertain you as well, because he's going to show real work in the field. So Emilio, the mic is yours.
Thank you, Spyros. I'm going to try to entertain everyone. As a huge number of attendants, what I would like to say good morning for some of you, or good afternoon, or really early in the morning, because not only from all around Europe, there are a lot of friends I can see in the list, but I would like to also give my warm welcome to my friends and colleagues from over the seas, from the Ohio State University. It's helping through, and Erdal Özkan here together with Carla. So really appreciate your attendance here. As you can see, not only in Europe, they are interested at what we are doing here in the whole continent. I hope I will be at the same level of excellence that my previous colleagues, Gerrit and Dimitris, they made an excellent job. And just before to start sharing my screen, I would like to really give my thanks to Spyros, not only for this invitation to be here showing you what we did, what we tried to do in this very difficult period within Optima, but especially for giving me the opportunity to be involved in this very amazing topic of the European projects. I'm talking from years ago. It's thanks to Spyros that I had the opportunity to experience that, and it's really amazing. So thanks a lot.
No, we thank you, Emilio. Anyway, go ahead, please.
Let me share the screen. I hope you are already able to see my presentation, right?
Yes.
Yeah.
Now it's great, please. Yeah.
Okay, thank you. So let me talk about the last part of the chain. So because sooner or later, farmers, they must use pesticides, and they need to do in a proper way. And then that's the reason why we have been working in a lot of years and try to develop new technologies, try to develop what we call smart sprayers, and try to help the farmers in the use. So this is the title, the importance of smart sprayers to improve crop rotation in general. I'm Emilio Gil, professor of agriculture school in Barcelona, Polytechnic University of Catalonia. I'm the director or main responsible of this unit of agriculture machinery research group. We are working mainly focused in general in all kind of agriculture machinery, but we're specifically in spray application technology since years. No, in my case, it's since 32 years I'm working in this topic. What we tried to do within Optima project was two main activities. One is linked to Work Package 4, which was try to develop together with our colleagues, especially with the spray manufacturers, try to develop this smart sprayer only for apples, in parallel with the same activity that our colleagues from Italy, my friend Paolo Vasari and his team, they have been working in vineyards, and Jean-Paul, my friend from INRA in France, they have been working in carrots. So our activity as a unit of agriculture machinery at the UPC has been try to develop this smart sprayer for apples. But also, we have been responsible of Work Package 5. And the main objective of Work Package 5 has been to test or to validate the results of our project. You saw already the amazing activity carried out by Gerrit and his team in Wageningen. But we, as Work Package 5, we try to evaluate in the field together with the other smart sprayers. So just a couple of words about who we are in OPC team, and give my thanks to all my team, especially to my young researchers, PhD students from all over the world, and all together we try to do our best. So, coming back to crop protection, all of you know that crop protection is not a simple task. There are a lot of aspects involved in this. So weather conditions, we need to know something about target. It's a tree, it's a small tree, it's a big tree. So what kind of product in this Optima project we have been working, as you have been able to see already, differences between synthetic products and bio PPPs and so on. What kind of a sprayer are we going to use? And in the top of them, we have the operator, we have the farmer, we have the tractor driver who must be able to use all the devices that we have been able to develop. So as nobody is perfect, nobody knows everything, at least me, I don't know nothing except some small things. I would like to give some words about those three main aspects. The target itself, so the canopy, in this case, focused in apples as our main responsibility. The equipment, which are the main characteristics of this sprayer or this smart sprayer, and in the top, the operator, who is in charge to use this sprayer and is the main responsible of the success at the end. All of you know already that Europe started years ago in a huge change in the legislation, in the rules concerning the crop protection, and in particular, the use of pesticides. No, we started in 2012 with the publication of Sustainable Use Directive, also entry in force the European directive concerning to new sprayers prior to be placed into the market. And now recently, we have the Green Deal, and we have specifically the farm to fork strategy. I took just some images from internet about the farm to fork. As it's plenty of challenges in this farm to fork strategy. Some of them easily or not to arrive, and some of those not so easy to arrive. Where is Optima project linked with this farm to fork strategy? Obviously, it's in the first part, among others, right? But in the first part, or at least what concerns to our activity, is linked to how can we reduce by 50% the use and risk of chemical pesticides by 2030? It's not an easy task, right? So we cannot go to the farmer and tell them that you must close one of each other nozzle. But to reduce the 50%, there is no sense. We must do something else. It's a huge challenge that we must combine all the tools that we have. But let me tell you that after more than 30 years working in this topic, I can tell you that more or less we can arrive to this figure. We can improve the situation. We can guarantee a more sustainable, and we can guarantee a more healthy food production. That farmers need support coming from the technology, but coming from other topics, as I will give you. Again, precision farming or precision agriculture. We have already an official definition of precision agriculture, right? But I use some other words, technology and communication as a base tools for a more productive, sustainable, and more competitive agriculture. And this is what the farmers want. More with less, this is what farmers want, and this is what European Commission wants. But it's easy to write and also easy to apply, right? And this is precision farming or smart farming or agriculture 4.0 topics and topics and topics. But we need to go something in detail and to help the end users in the use of this technology that we have already, this is already available, but in some cases, in most of the cases, end users has not the opportunity to deal with, to use and to take benefits. Then the objective, specific objective of this development of the smart sprayers in Optima was that exactly this developed this variable rate application sprayer, in this case for apple trees, trying to achieve on a time variable optimal adjustment of pesticide according to the canopy characteristics. Just let me show you 3 different pictures of the same apple plantation here in Spain. Obviously, the target is completely different from the left to the right side. Obviously, the ideal situation will be that the sprayer could be able to apply according the canopy, according the differences in the canopy and according what they need exactly. It's again easy to put some pictures, but not so easy because there are some other aspects linked to that and not linked to the technology that I would like to underline or mention later. And right, let me show you this picture we made years ago when we tried to help this farmer in the spray application in his peaches in Aragon more than 10 years ago. It's a real picture, unfortunately. It's not a Photoshop picture. And I can tell you that after the calibration, the sprayer adjustment, and the air adjustment of this sprayer, the farmer was really happy because we were able to show him that he could reduce. I don't remember the amount of pesticide, but if you can take a look of this cloud, and if we are able to reduce this cloud, easily we can arrive to the 50% of reduction according to the farm to fork strategy. So PPPs can be reduced. We have tools to reduce. We have technologies and we have training. But after our experience, when we merge together training plus technology, we have a high success. We have high possibilities to achieve these challenges already written in the Green Deal and in the Farm to Fork strategy and in the new CAP, in the new Common Agriculture Practice, because it's explicitly written that we must implement new technologies, we must implement precision farming. But not only precision farming, it doesn't work without very well educated farmers. And one of the aspects I would like to put on the table, just before to continue with the technology, is linked to, let's say, legislation or some not really technical issues. Is linked with the PPP dose expression. If you take a pesticide label, in most of the cases, pesticide dose is based in kilos per hectare or in concentration. Just let me give you this, put this question over the table. Left and right side of the picture, same plantation, same area, 1 hectare on each case. If the pesticide level gives me the recommendation in kilos per hectare, there is no sense because I have 3, 4, or 5 times more leaves in the right side than in the left side. So this is something to be at least analyzed. What kind of recommendations are included already right now in the pesticide labels? It's a huge discussion, but just some of you or all of you have in your minds the recommendation based in 1,000 liters per hectare. Nobody right now is using 1,000 liters per hectare. It's against the technology and against the OPTIMA developments we are working. But having that in mind, we can easily demonstrate that with a well-adapted sprayer, with a well-calibrated sprayer, and using alternative pesticide dose recommendations, we can easily be able to reduce the amount of pesticide. We can easily reduce the amount of applied water in this case, and we can increase the working capacity of the sprayers. And the farmers can do that in several ways. Farmers can do that manually. If we provide them, as we already are doing, good indications, good guidance, how to measure their canopies, how to estimate the target they need to spray. And if we provide them tools or practical tools to help them in the canopy characterization, to help them in the calibration process, easily they will realize that they can reduce the amount of pesticide, that they can increase the economical benefits, because at the end, farmers want to control the pests, and they want to control the diseases, and if they can reduce 10%, 50%, or 20% of pesticide, it's absolutely welcome for them. But first objective is to control pests and diseases. By the way, we have plenty of already available material, simple material. We don't need to invest in a satellite. We don't need to invest in 1,000 euros in a GPS or whatever. Practical material, as the couple of examples here in the screen, are already available for farmers. And this is what we have been working in the last 4 years, trying to collect all this material already available, put it in a kind of certain order, classify by thematics, by type of materials and so on. I'm very glad to that have been coordinator of this InnoSeta project. And within this InnoSeta project, we have been able to develop this InnoSeta platform, plenty of material already available for the end users in 8 different languages. So no excuse for don't use this very, let's say, simple material that will help the farmers within InnoSeta. But this is if farmers wants to do in a manual way. But what we did, what we are doing in OPTIMA project, it could be nice and ideal if the sprayer could be able to measure by itself the canopy and decide how many liters of water and how many liters of pesticide would be able to apply. This is on the left side of the screen. You can see the map. This is a canopy map, a real canopy map already implemented by this Apple Smart sprayer in the region where we arranged the field trial. It was in Épila region in Zaragoza here in Spain. And this is in general the idea, what we wanted the idea. Try to adapt the amount of pesticide, the amount of liquid according to the canopy characteristics. So we started, in fact, we started with this principle more than 20 years ago in my research group, installing, in this case, we decided to install ultrasonic sensors as we compare the price with another sensors, onboard sensors are really cheap sensors. Of course, there are some difficulties, some aspects that can be compared with some other sensors as leader and not so good, but we decided at the end to go through these ultrasonic sensors and try to develop some kind of, let's say, software in order to characterize as much as possible the canopy, and then, by the way, with this information, transform this information in the optimal volume rate. Of course, we already developed the fundamentals based on the information provided by the ultrasonic sensors. We made a huge work. I would like to especially thanks to my students, Bernat, who is making his PhD. He has spent a lot of time in the development in the electronics and calibrating ultrasonic sensors, calibrating pressure sensors, calibrating everything, then implementing the hardware into the sprayer, mixing the hydraulic liquid with the electronic liquid. So we decided to work with using these Raspberry Pi models with specific ultrasonic sensor. It was difficult to find the correct proportional electro valve and the flow sensors and so on and so on. Let me tell you that I'm not expert in electronics. So I had the idea, but then it's quite difficult to arrive to the final solution. Then this is, you can see some pictures. We had support from Pulverizadores Fede in this case, not only giving us the sprayer, but giving us the practical support in building this structure, where to put the ultrasonic sensors covering the whole canopy, the canopy area. Also providing us with the practical material for installing the 6 electrovalves, because the sprayer is able to control in 6 different sections, 3 in the left side and 3 in the right side. And you can see also there detail the box, plenty of electronics, that thanks again to the huge work from my PhD students. Then we arranged preliminary test in our facilities in Agropolis in Viladecans. We have some grapes there, but we have not apple trees. So we decided to create a new apple trees plantation, in this case, artificial trees. It's a good point because we can manage the leaf area. We can decide how many leaves per square meter or per cubic meter. And we arranged thousands of tests there, trying to calibrate the ultrasonic sensors and try to combine the information provided by the ultrasonic sensors and define 2 main aspects in the canopy. The canopy width at the 3 different levels, but not only the canopy width, but also the canopy density by a huge and tedious, let's say, mathematical calculation and estimation, right? So at the end, we have been able to obtain those main parameters. Also, we measured, we evaluated with the digital images this canopy, this artificial canopies, and linked it with this information with the signals, the 1,000 values of the signals obtained from the ultrasonic sensors. We arrived to a conclusion. It will be really positive or not, or we will be true or not, but it was the conclusion that we arrived at, and at the end we have been able to characterize this canopy. Then, with our experience, with our 20 years of experience, it's not enough having information about the canopy. What we need to do is how many liters per cubic meter or how many liters per square meter of leaves we need to apply. And this is something, let's say, the agronomical knowledge. It means that there is not a single smart sprayer in the world able to apply by itself. It's still needed this agronomic information. Once the sprayer was already developed and tested in our facilities, considering all the difficulties linked with COVID, impossibility to travel, difficulties to go to the field, so as you guys already suffer as me, but it has been really one of the most difficult parts. Then finally on May 2001, we were able to arrive to Epila in Zaragoza, and we started to evaluate this smart sprayer among the other developments in the project. And we compare with the other two sprayers. The first one, we call it the conventional one, is as the farmers use the sprayer. And the second one is this another farmer, but following this best management practices. So a well-calibrated sprayer, well adjusted in the air, the working pressure, forward speed, and so on. And the last part was the OPTIMA smart sprayer. And we already arranged trials for the position in 2 different periods, in May and in November. And as you can see here, this is the average, just a simple value. I don't want to bore you with detailed analysis and statistical, but this is the deposit in micrograms per square centimeter in conventional sprayer, in the well-adapted sprayer, and with the OPTIMA smart sprayer in May and in November. In all the cases, no statistical differences. But the most important is, if you can take a look at the figures here, the farmer was applying close to 900 liters per hectare. With the reference one, with the best management practices sprayer, we adjusted at 700, less than 800 liters per hectare. And with a prototype, in both cases, in May and in November, the total amount applied was less than 500 liters per hectare. So it means that if we maintain the concentration in the tank, what we did, we have been able to reduce by close to 50%, 45, 47%, the amount of liquid, and at the same time, the amount of pesticide. And we have data because we already made biological efficacy control. Farmer has been able to guarantee the same disease and the same pest control. So we have been able to achieve more or less recommendations established by the farm to fork strategy. Of course, considering the sustainability, environmental aspects, and so on, within the project, we agreed to make different drift measurements, trying to follow this standard 22866, which is really terrible to follow, but we did it. Thanks to my PhD student, Lu Chun from China. He's a real clever guy, and we were able to do that. And let me show you just a short overview. This is a drift curve with the 3 sprayers, and the yellow one corresponds to the smart sprayer. In all the cases, drift was reduced considerably in comparison with the conventional application used by the farmer. So not only in the position, but only in drift, and we have been able to reduce, let's say, 40% of product. I think this is the objective. Of course, it's not everything is not everything is happy. Not everything is happiness. We had a lot of technical difficulties with certain products. We had difficulties with the electroval. We had difficulties, plenty of difficulties. That's the reason why we will continue working in this next year. And another aspect I would like to mention, remember the excellent presentation by my colleague Gerrit in World Package. They developed this nice device, a spectacular device. We install it in our system here in our tractor with the camera, with the screen. And what we did in one of the parcels, in this parcel, we used this ETS. We detected the Apple scab in this case, and we were able to identify the areas where the Apple scab was present. And what we did was to merge the variable rate application sprayer with this disease map. In general, in this parcel, it was not so big parcel. For conventional application, the farmer will use it to apply 250 liters in total. With the variable rate application, we applied less than 200. And when we merged variable rate application with EDS, we reduce it up to 44 liters in this area and in this area. Of course, this is just a preliminary activities, but this is another example about what we intended to do within the OPTIMA project, trying to merge all the developments, the DSS, the EDS, and the smart sprayer. But what I would like to give you is this 30 seconds video, which is the opinion, some impressions of one of the collaborative farmers in Épila. This farmer, José Antonio, has been using the smart sprayer during the whole season last year. And just listen to him better than me.
To guarantee an adequate control of apple scab.
Con el atomizador, con los sensores que lleva, reduces la cantidad de fitosanitario, bueno, la reduces, no, la ajustas al nivel de vegetación que tú tienes. Por lo tanto, aplicas justo el producto que necesitas. Ni más ni menos, como suele pasar. Si aplicas menos, no controlas la enfermedad, tienes que repetir, por lo tanto es doble gasto, doble producto, y si aplicas más, es mal empleado, es tira.
The results that
So, in my opinion, this is the best impact factor we can demonstrate. So farmer is really happy with us. In fact, we will continue working in the next season. And then I think I have 5 more minutes because I would like to show you the real images, what we did during this season. We already produced this 4.5 minutes click video. It's already in OPTIMA website. It's already in the European Commission website. And with this one, I would like to thanks to all of you for your passion, your attention, and just try to enjoy the video.
Precision agriculture is already presently here. This prototype, developed within the framework of the OPTIMA European project within the H2020 program, is capable of adjusting in real time the amount of pesticides depending on the changes on canopy characteristics. This smart sprayer has been developed with the aim to improve the profitability and sustainability of farms in apple production. Based on the experience after many years of work, the Agricultural Mechanization Research Team from Polytechnic University of Catalonia, one of the partners of the OPTIMA project, started to develop the theoretical principles of a new sprayer capable of continuously modifying the amount of liquid applied during the pesticide application process, based on the specific characteristics of the trees. The use of ultrasonic sensors allows obtaining precise and detailed canopy characterization. This information is then analyzed and transformed into important canopy dimensions as canopy width and canopy density, required parameters to establish the optimal amount of liquid and pesticide to be applied. The process is carried out in real time, while the sprayer circulates between the rows of the plantation. The thousands of data obtained by the ultrasonic sensors are automatically analyzed in a dizzying way thanks to specific hardware developed by researchers at the Polytechnic University of Catalonia. This data analysis includes to convert every few seconds the electrical signal emitted by the ultrasonic sensors into the calculated canopy dimensions. Then the process continues, always within the same time lapse, by transforming the already generated data of canopy into the optimal volume rate to be sprayed by applying the agronomic experience. Once the optimal dose is calculated, the next step is to transform this value into the electrical signal to be provided to all the 6 individual electrode valves in charge to modify the individual nozzle flow rate. All the required steps to accomplish the process are controlled by the dedicated software developed by the Polytechnic University of Catalonia.
Hemos conseguido que esas boquillas que están ahí en la máquina cambien el funcionamiento. En un momento echen 1,5 litros y otro medio litro y en otro 1 litro por minuto, en función de lo que está viendo en tiempo real.
The nozzle flow rate of every single nozzle is automatically adjusted according to the optimal established pesticide dose. Within a very short time, every single nozzle is capable of regulating the emitted flow rate. A novel technique that the fruit growers of Epila in the Spanish region of Aragon have tested and analyzed during one full season. Epila produces 11% of the Spanish apple production. With the help of this smart sprayer, farmers have been able to reduce the amount of pesticide during their daily work, without affecting the quality of the fruit. Thanks to the developed system, they can adjust the amount of liquid to be sprayed according to the canopy characteristics. At the same time, drift losses have been reduced and the spray quality has been improved compared to traditional system. Results of the OPTIMA project are totally aligned with the requirements of the recently published European strategy from farm to fork, where an important challenge of 50% of risk reduction during pesticide application has been established.
So that's all from my side. Thank you for your attention.
Emilio, I think that Spyros is he was thrown out of the meeting, so
I'm back. Sorry for that.
He's back.
You're back. So
I think I apologize.
Thank you.
Yes. Thank you, Spyros.
Thank you, Thanos, for helping. So again, just to close, apologize for this. Emilio, as I said, we expect it to be a very entertaining and very informative, of course, presentation. It's very impressive how much, I mean, you have really targeted the core of Farm to Fork Green Deal, that we can, just initial work that we can manage. We actually managed to reduce 50% of the PPPs, and also together with the vision system that Gerrit, that was impressive, of 150 liters to go down to 40. So that's impressive. So thank you very much. So any questions maybe from the audience? If anybody would like to ask something?
They are hungry.
Okay, if not, then we can have a discussion, I think, with also, we have 10 minutes before we close, with also with Gerrit and Dimitris. Please also, you can come on board and present yourself, your camera. Because I have the question, for example, because in Optima, we have offered to have a holistic IPM system. That means how are you going to incorporate also the IPM strategy? For you said, Emilio, that you're going to also work this period, this growing season, May and June, right, for apples. So are you going to incorporate also the suggestions by Dimitris for which pesticide?
Of course, we must. No? In fact, this is one of the activities we already did in the last season, using a combination of synthetics and bio-PPPs. Unfortunately for the project, but fortunately for the farmers, in our case, in Apples Cup was really a low pressure this season. But even though we had already some results, and we will continue working.
So Dimitris, I think you will be able this year to give this recommendation, the IPM strategy, right, with a combination of synthetic and bio-PPPs for the three.
Yes. We're expecting also to see the results from the pilot testing in all the countries, and then we can all discuss together and end up with a schedule of spraying schedule based.
Excellent. Yeah, that's very nice. So I think we don't see any more questions. Actually, say that our project officer, Francisca, was following all the discussion, all the presentations. She just left a few minutes ago. She also would like to congratulate all of you with great presentation. She said she's got many more questions for the final review meeting based on this. She also liked the cats and dogs of Gerrit. She could understand more what the artificial intelligence is about, what she mentioned. So yes, I think with this, we would like then to have a final word from each of you, and then we can close. Dimitri, just a final word of your experience so far, and what do you expect for the next 6 months, and also for the audience.
Yeah, I think that the results are really amazing. You can see, and I think the overall goal was basically to reduce as much as possible the spraying, that's the spraying drift, and the amount that we spray in our orchards and fields. And I think with the combination with the right IPM system, I think we can manage to target, basically to control quite efficiently the disease and have also a very huge environmental impact.
Great, thank you very much, Dimitris. And Gerrit, a few concluding remarks, please.
Yeah, for me, it was a real pleasure to work in this Optima project. I liked it very much to cooperate with all the partners all over Europe. We had a lot of interactions. We visited the fields in Italy and in France. Unfortunately, not in Spain yet, but hopefully we can do it this year if COVID is getting a bit better. We got a lot of help also from the partners. For us, it's very important to have good annotations in the images. And we need real crop experts there. And we got a lot of help there from the partners, so that was very helpful. The plan for us is to continue to work on the spectral imaging. We expect that we get for the grapes, for the apples and the carrots, that we can increase the results quite a bit. So we will continue to work on that. Yeah, that's mainly my plan.
Great, thank you, Gerrit. We're expecting the higher correlations in the end of the season. I think that's a challenge, but good challenge. And then finally, to Emilio, to say a little bit about more with less, to expand this nice, let's say, slogan.
Yes, as I already mentioned during my presentation, we must combine technology with education, with training, because when you are in front of this spray, it is quite difficult just to turn on the tractor and turn on the spray. So we need a very well-trained farmer. So that's the reason why I took the opportunity to remember what we did. You were involved as well in the Noseta project. And by the way, absolutely agree with Gerrit. It is a real pleasure to work with this OPTIMA team. We learn a lot, because it's really amazing when we were in the field, because we are experts in the sprayers, but not in the cameras trying to identify diseases. And it was funny working with this, and over the cell phone, Peter in Netherlands, and we were explaining him how was the apple trees look at that. So, and at the end, I'm really happy to see how farmers are really interested in the new technologies, in the development, how they really appreciate, because it's quite difficult to find farmers able to be there during the whole season. Now I need to change the tractor, now I need to do this, now I need to... At the end, they are happy. So in this sense, this is a message for the European Commission. We have been able to arrive to the end users.
I think we couldn't conclude with the best, let's say, keywords that the farmer was happy, and that's the most important. So that's why we feel that we have contributed something. Each project contributing like a piece, but we have contributed our piece, I think, over the last 3 years, and we still wait for the next period. So again, I would like to thank very much the panelists. You were excellent. Thank you for your work. You are like amazing people, also very collaborative, very nice to work with. We all work together as a team. And I'd like to thank everybody for attending this meeting, and please try to attend our social media and all the news and the videos. So thank you very much again for the panelists first and all the audience. Bye-bye.
Thank you.
Thank you. Bye-bye.
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 Bye-bye.